Dear students, we are going to discuss Dioxic culture and Synchronous culture. These are two different types of growth patterns observed in bacterial cultures. What is a Dioxic culture? It is the diphasic response of a microbial culture to the addition of a second substrate. So, when certain microorganisms are grown in the presence of two different nutritional substrates, then they show this type of a growth response, which is known as Dioxic culture. Such bacteria will show two phases of growth. They will have two growth phases or two growth peaks. This is shown by bacteria which are having inducible enzymes. Inducible enzymes are produced only when it is required. For example, some bacteria, when they are grown in the presence of glucose, which is an easily assimilable nutrient source, and lactose, which need to be cleaved or hydrolyzed to glucose before utilization. If bacteria are grown in the presence of both glucose and lactose, then they will be growing by using glucose first. After the depletion of glucose only, the bacteria will produce the enzymes required for lactose metabolism, for utilization of lactose. So, such bacteria which are having such inducible enzyme systems, they are generally showing this type of dioxic culture pattern. So, in this case of such bacteria, when it is grown in the presence of both glucose and lactose, there will be two growth phases. The first phase, when they are growing by using glucose, followed by the growth phase during which the bacteria are utilizing the less common sugar, that is lactose. So there are two lag phases and two logarithmic phases. The first lag phase and the first logarithmic phase are during when the bacteria are growing by utilizing glucose, and after this first logarithmic phase there is a brief delay during which the enzymes required for lactose utilization are synthesized. So this is the second lag phase during which the bacteria are getting adapted to grow by using lactose, and once the inducible enzymes are produced, now the bacteria grow through the second logarithmic phase during which they are growing by using the second less common sugar or the second substrate, that is lactose. Such a two-phase growth is known as a dioxic culture. There are two phases or two growth peaks, and such a bacterial growth pattern is known as dioxic growth, and we will get a dioxic growth curve when such a dioxic growth is plotted. The growth pattern or the growth population size, when it is plotted against time, then we will get a dioxic growth curve. It is a growth curve generated from a dioxic growth, from a dioxic culture. Now we will discuss a typical dioxic growth pattern. It could be obtained but when a bacterium is grown in a media which contains very low concentrations of glucose. The glucose is the first substrate here, and the media will be containing lactose also. So when a bacteria is grown in a media containing both glucose and lactose, we will get a typical dioxic growth pattern which have two distinct phases of active growth. And when such a growth pattern is plotted by plotting the population size on y-axis and the time in the x-axis, then we will get a dioxic growth curve which have two lag phases and two logarithmic phases. So in this image we can see that there are two lag phases and two log phases. During the first lag phase, the bacterium is getting adapted to the new cultural conditions, and during the logarithmic phase, the bacteria will be growing by utilizing glucose. So during the period of glucose utilization, lactose is not at all utilized because now the cells are not able to transport and cleave the disaccharide lactose. The more simpler form of sugar, that is glucose, is utilized. If glucose is available, then it is always metabolized first in preference to other sugars. Only after the complete utilization of glucose, then only the metabolism or the utilization of lactose will begin. So from this image, which is the dioxic growth curve of Escherichia coli grown in a limiting concentrations of glucose and lactose, during the first lag phase and the first exponential or logarithmic phase, the bacteria are utilizing glucose only. So after some time, due to the growth of the bacteria, the glucose will be completely utilized, and then the bacteria will start synthesizing enzymes required for lactose utilization. In the presence of glucose, this particular enzymes required for the lactose synthesis, which are controlled by a gene regulatory mechanism known as lactose operon. In the presence of glucose, this Lac Operon is repressed. But when the glucose concentrations go down, when the glucose levels are depleted, then the Lac Operon will be switched on and the enzymes required for lactose metabolism will be synthesized. And this is the phase, the second lag phase. And during the second lag phase, now the bacteria are starting to synthesize the enzymes required for lactose metabolism. The bacteria are getting adapted to grow by using lactose during the second lag phase. And during the second exponential phase, now the inducible enzymes, the enzymes required for the lactose metabolism are completely synthesized, and now the bacteria start to grow in an exponential pattern by using lactose. So this is a typical Dioxic growth curve which is shown by bacteria when grown in the presence of glucose and lactose. During the first lag phase and first logarithmic phase, the bacteria are growing by using the more simpler form of sugar, that is glucose. And upon the depletion of glucose, then the bacteria will start to synthesize the enzymes required for lactose metabolism. And there comes the second lag phase. And after the lactose utilization enzymes, they are completely synthesized, then the bacteria will grow during their second exponential growth phase, during which they are utilizing lactose. When bacteria are grown in a media which contain both glucose and lactose, then the microorganisms will be metabolizing glucose first. Lactose is not utilized because the enzymes required for lactose metabolism are not present always within the cell. They are inducible enzymes and they will be produced only when it is necessary. So glucose is always metabolized first in preference to other sugars. Only after glucose is completely utilized, the other sugar such as lactose will be utilized. So the lactose operon, the Lac Operon, which is the regulatory mechanism by which the synthesis of the enzymes required for lactose utilization are controlled, the Lac Operon will be inhibited or will be repressed even in the presence of lactose if there is enough glucose is available. So glucose is a better source of energy than lactose, since for the utilization of lactose, two additional enzymes are to be synthesized. So the bacteria will go for the utilization of glucose if there is glucose present, and only after the complete utilization of glucose, the Lac Operon will be initiated in the presence of lactose. So the Lac Operon will be switched on only if the glucose levels are exhausted. Then the lactose molecules can act as inducer to switch on the Lac Operon, and as a result, the enzymes required for lactose utilization will be synthesized. So even if the lactose, the inducer molecule is present, the Lac Operon will be repressed in the presence of glucose. This phenomena, that is, the availability of glucose, inhibits or represses the synthesis of enzymes required for lactose utilization, is known as catabolite repression or glucose effect. The ecological rationale for this is that glucose is a better source of energy than lactose, since the utilization of glucose requires 2 less enzymes. So, only after the complete utilization of glucose, then only the bacteria will move on to switch on the synthesis of enzymes required for utilizing lactose. This is known as catabolite repression or glucose effect. Only after glucose is exhausted, the enzymes for lactose utilization are synthesized. So, these enzymes for lactose utilization are synthesized during the secondary lag phase during the Dioxic growth. It is the time required for the complete induction of the Lac Operon and the synthesis of enzymes necessary for lactose utilization. There are 2 enzymes required for the utilization of lactose. They are lactose permease and beta galactosidase. So only after this second lag phase during which these enzymes are synthesized, then only the growth of bacteria occur, during which they are using lactose, since up to that time the bacteria were growing by using glucose. So after the exhaustion of glucose, the bacteria will switch on the Lac Operon. They will synthesize the enzymes necessary for lactose utilization, and then they grow by using lactose. So the availability of glucose represses the enzymes for lactose utilization even in the presence of lactose. If both glucose and lactose are present in the media, the bacteria have a preference towards glucose. So this is known as catabolite repression or glucose effect. So if both glucose and lactose are present, the bacteria prefers glucose, and let us look into the mechanisms by which such a selection or such a regulation of metabolism is possible. So in the presence of glucose, the activity of a particular enzyme, adenylate cyclase, is blocked. Since the adenylate cyclase enzyme activity is required for the synthesis of cyclic AMP from ATP, the cyclic AMP is not synthesized in the presence of glucose. So in the presence of glucose, no cyclic AMP synthesis is there. The cyclic AMP is in turn required for the initiation of the Lac Operon transcription, since cyclic AMP is required to activate protein known as catabolite activator protein. This catabolite activator protein is required to stimulate the binding of RNA polymerase onto the promoter of Lac Operon. So, if there is no catabolite activator protein, then the RNA polymerase cannot bind to the promoter site in Lac Operon, and as a result, the Lac Operon will not be transcribed. There is no initiation of transcription of the Lac Operon. This happens in the presence of glucose. In the presence of glucose, adenylate cyclase activity is blocked. As a result, there is no cyclic AMP. So, there is no activation of catabolite activator protein CAP, and as a result, the RNA polymerase cannot bind to the Lac Operon promoter. So, the Lac Operon is not activated, it is not initiated. There is no transcription of the Lac Operon in the presence of glucose. While in the absence of glucose, if there is no glucose available, then the adenylate cyclase activity is very high. As a result, the adenylate cyclase will produce cyclic AMP molecules from ATP. In the presence of cyclic AMP, the catabolite activator proteins are activated. The catabolite activator protein will help the RNA polymerase to bind to the promoter site in Lac Operon, and as a result, the Lac Operon will be transcribed. The transcription of the Lac Operon will start in the absence of glucose, but there is an additional condition required for the transcription of Lac Operon, that is, it requires the presence of lactose as well. In the absence of glucose, cyclic KMP are available to activate the catabolite activator protein, which will allow the RNA polymerase to bind to the promoter. But for the transcription of the Lac Operon to begin, there is an additional requirement, that is the inducer molecule for the Lac Operon, which is lactose. The lactose has to be present. Only in the presence of the inducer molecule, then only the Lac Operon will be initiated. So, in the absence of glucose, the RNA polymerase will readily bind to the promoter site in the Lac Operon, and if lactose is present, it will act as an inducer, and the binding of the inducer is also required for initiating the Lac Operon transcription. Thus, to efficiently promote transcription of the Lac Operon, lactose must be present. Then only the lactose will be able to inactivate a particular repressor molecule. A Lac repressor is there. It will be inactivated only in the presence of lactose. So there are two conditions required for the initiation of Lac Operon. There should be no glucose. There should be absence of glucose, and also there should be enough lactose available. Then only the RNA polymerase will bind to the Lac Operon promoter, and the lactose molecules, the inducer molecules, will bind and inactivate the repressor molecules, and then only the transcription initiation of the Lac Operon will begin. So this is the mechanism by which a bacteria manages to grow in the presence of two sugars, two molecules, that is glucose and lactose. If there are glucose available, then the bacteria will utilize glucose first, and only after the complete glucose utilization, the bacteria will switch on to utilize lactose. So for the utilization of lactose, certain enzymes are to be synthesized. It is an inducible operon, the Lac Operon, which will be activated to synthesize the enzymes required for lactose metabolism if there are two conditions met. The first condition is there should be no glucose available. There should not be any glucose available. The absence of glucose is the first factor. And the second factor is that lactose must be available. So only in the presence of lactose and in the absence of glucose, the Lac Operon will be activated, and the enzymes required for lactose metabolism will be synthesized, and then only the bacteria will be able to grow by utilizing lactose. So, in the presence of glucose and lactose in the culture media, the bacteria will show a typical growth curve in which there are two growth phases, a first lag phase and a first exponential phase, during which the bacteria utilize glucose, and a second lag phase and a second exponential phase, during which the bacteria use lactose. So this is Dioxic growth and a Dioxic growth curve. Now we will discuss synchronous or synchronized culture. Generally, in any bacterial culture, there will be cells growing at different rates, and as a result, there will be cells in all stages of the cell cycle. But in a synchronous or a synchronized culture, this is a special type of a culture which contains cells, all cells in the same growth stage. The cells all will be in the same growth stage in a synchronous culture. Obtaining such a culture, in which there is a unified cell cycle stage that is very useful for biological research. For research purposes, since the cells are too small, certain techniques require this type of a synchronous culture, which could be treated as a single cell, since all the cells are growing in the same rate and during the same stage. So, the synchronous cultures have been extensively used to study cell cycle, cell growth, and also the effects of various factors on cell cycle and cell growth. So, synchronous culture is a type of culture in which all the bacterial cells are in the same stage of their growth, and as a result, the growth curve of a synchronous culture will be showing a ladder-like pattern since all the cells are dividing exactly at the same time. So if it is a culture of E. coli cells in which the generation time is about 20 minutes, at every 20 minutes we can see a doubling of the population since all the cells are dividing exactly together. We can see each generation in a synchronous culture is the next step in a ladder. The synchronous culture will show a ladder-like growth curve. In this image, we can see the non-synchronous growth in a curved pattern, while the synchronous growth is showing a ladder-like pattern. Here, the number of generations is plotted in the x-axis and the number of bacteria, the population size, is plotted in the y-axis. The ladder-like growth pattern is shown by a synchronously growing culture of bacteria, while the non-synchronous growth is shown in a curved-like appearance. Generally, under natural conditions as well as in normal laboratory conditions, the bacterial cells will be growing in a non-synchronous pattern. The cells will be growing and replicating at different time points. Even though if they are growing in a normal growth rate, the cells will be dividing at different time points. So to obtain a synchronous culture, we have to do certain techniques. So to obtain synchronous cultures, we may change the external growth conditions to arrest the growth of all the cells in a culture, and we can resume their growth together by reversing the conditions. As a result, the newly growing cells will be starting to grow at the same stage, and then we will get a synchronous culture. So there are several techniques to obtain synchronous culture. They are providing limiting conditions for some time, the addition of growth inhibitors for some time. We can perform size dependent sorting to obtain inoculum for obtaining a synchronous culture. And a technique is there known as Helm Stator Cummings technique. So what is the first technique? That is providing limiting conditions for some time. So here we will not provide the necessary growth conditions for the culture for some time. For the case of photosynthetic bacteria, we will not be providing enough light. The light will be eliminated for several hours, and then when light is reintroduced, then all the cells will grow together. They will start to grow together and they will be growing at the same stage. Another method is starvation, not by providing enough nutrients. So generally phosphate is limited. The phosphate limitation will inhibit the growth of the cells in the medium, and when we reintroduce phosphate to the culture medium, once the limiting factor is reintroduced, all the cells will start to grow together and will grow in the same phase, and they will give us a synchronous culture. So this is providing limiting conditions for some time, and when the limiting conditions are reversed, when we provide the necessary requirements, then the cells will be growing and providing a synchronous type of growth. The second technique is the addition of growth inhibitors for some time. So here To inhibit the growth of microorganisms, we are adding certain chemical inhibitors. The addition of chemical growth inhibitor will stop the growth of all the cells. Then we can remove the inhibitor, and the removal of the inhibitor will initiate the growth of the cells, and the cells will grow together and they will begin to grow synchronously. An example of such a growth inhibitor is nocodazole, which is often used in biological research. The addition of nocodazole will inhibit the microbial the cellular growth, and when we remove the nocodazole completely from the medium, then the microbial cells will start growing in a synchronous pattern. But this technique, the addition of growth inhibitors, have a disadvantage that to obtain the synchronous growth, to initiate the synchronous growth, we have to completely remove the particular chemical growth inhibitor, which is a very difficult procedure. Another technique to obtain synchronous culture is size-dependent sorting. So, in a bacterial culture, if the cells are growing at different stages, the cells in different growth stages, they will have different physical properties. So, they can be separated, physically separated based on their density or size. We can use centrifugation for density-based separation, or for size-dependent separation, we can use filtration. So, the population of the cells can be fractionated based upon their size or density, and the cells having the same density or the same size, those cells can be used as an inoculum to initiate a fresh culture in which they will be growing in a synchronous pattern. We will get a synchronous culture if we use the cells of a same density or same size as inoculum. So, either we can use the smaller cells having smaller size, or smaller cells having low density, or larger cells having larger density can be used as an inoculum to obtain a synchronous culture. Next is the Helm Stator Cummings technique. This is also a filtration based technique. Here, a bacterial culture is filtered through a membrane. During this filtration, majority of the bacteria will pass through the membrane, but some will remain bound onto the filter membrane. When fresh medium is applied to this membrane, the bound bacteria, the bacteria which are bound onto the membrane, they will start to grow. The newly synthesized, the newly divided bacterial cells, they will detach from the membrane and can be collected from the bottom of the filter, from the apparatus, and can be grown as a synchronous culture. So, here a population of cells is passed through a membrane filter. Certain bacterial cells will be trapped in the filter. The filter is then inverted. Fresh nutrient medium is flowed through the membrane filter. Then the loosely associated bacteria are washed from the filter. Most bacteria will be passing through, but some will remain bound to the membrane. And if we provide fresh medium, these bound bacteria will start to grow, and the newly formed bacteria, the newborn bacteria, will detach from the membrane. They all will be in the same stage of growth, and they will start to divide in a synchronous manner. But this method have one disadvantage. The number of cells, the number of dividing bacteria which could be collected from the apparatus is very small. Very low number of bacterial cells will be grown. The population size will be very small if we use Helm Stator Cummings technique to obtain synchronous culture. So, these are the different techniques to obtain a synchronous culture. We can provide limiting conditions for some time, and once we reintroduce the growth conditions, then the cells will grow together in a synchronous pattern. If we add growth inhibitors for some time, upon the removal of the complete growth inhibitors from the medium, then the cells will start to grow in a synchronous pattern. We can perform size-dependent sorting either by use of centrifugation, density gradient centrifugation, or filtration for size dependent sorting can be done to obtain inoculum to start a synchronous culture, or we can use the Helm Stator Cummings technique to obtain a synchronous culture. So we discussed Dioxic culture and synchronous culture. Thank you so much for listening.